Okay, let's go ahead and get started. A very warm welcome to everybody to the second program of this week. Welcome back those who were here with us in the beginning, and welcome to the new people. My name is Olga Vitek. I'm an associate professor in the College of Computer and Information Sciences at Northeastern University. I'm a statistician by training, and our research is in all things quantitative mass spectrometry and proteomics, and I'm one of the co-organizers and co-hosts of the whole institute, so it's really a special pleasure for us to host this event here. Just as a show of hands, how many of you are coming here who were not with us for the last 2.5 days? A few. Okay, so let me just do a very quick introduction to some housekeeping items. So, as most of you know, a lot of the communication happens through Piazza. So Piazza is essentially an interface for communication, also for posting resources, answering questions, and things like that. So you all have received an invitation to that. If you do not know how to get there, please let us know maybe at the end of the hour and a half. The really important link on Piazza is here. So if you go under Resources tab on Piazza, there is the general information which has the website with all the schedule, Google Drive, this is where we post slides and handouts that you can copy. There is a YouTube channel, so we record both parallel classrooms, so you can actually watch the videos of the recordings. And there's also a link for the feedback survey. So in particular, if you want to ask instructors questions, so there is Q&A tab, and you can start. And so, for example, let's say I want to have a new post. So if I post new, so I can say I can post to the entire class, or I can post only to the group. Let's say here we have intro R. So this way you don't spam 100 people who are part of the event, but only write to those who are in this program. So this is really very, very briefly, right? And those of you who are new and who received travel fellowships, we have some forms for you to fill, so make sure you find us during the break as well. Okay, so that's in 2 words about the logistics. So now let me tell you a little bit the plan for the next 2.5 days. So this is a program for an introduction to statistics and introduction to R. So we will kind of take turns a little bit talking about statistical methods and then talking about the infrastructure in R, and in particular in RStudio. So this is a very, very introductory program. We really start very slowly with both statistics and R. Now for the statistics part, I will talk about things which are fairly general. They're not necessarily very specific to mass spectrometry and proteomics, although I will try to make examples which are relevant to that. And then starting Monday next week, 2.5 days, we'll build on what we discuss here, and we'll go specifically into tools for proteomics and working with the interface with tools such as Skyline and OpenMS and others as well. So that's the plan. So for the first hour and a half, I will start sharing with you some of the basic concepts of statistical design of experiments and also the analysis of the data. And if you have any questions, please make sure to stop me, because it's really, I hope that this will be kind of a conversation, right? More of an interactive discussion. Okay, so let's go ahead. So before I actually talk about all the statistical methods, I would actually like to share with you an experience that we had which motivates, in fact, this whole program, this whole 2 weeks, which was one of the motivations of why we need to create short courses or main institutes, and why this is so important. And this is another reason for why I would like to share with you that is because I will also introduce a data set that you will be working with as part of this 2.5 days and also 2.5 days next week. So, okay, let me first talk about that. So I was involved in the study by the Association of Biomedical Resource Facilities, and you probably know about that. So this is an association of essentially people who work in core facilities. So these are not necessarily people who try to develop new cutting-edge methods and develop methods which improve upon the state of the art, their main mission is to have well-run core facilities which can collect the data, acquire data, analyze the data, and provide meaningful results with well-established methods to their clients. And so this association often does studies which establish good practice. And for example, you can have biological material which is shared between different labs, and they all do the same type of steps, and then they compare the results. In this particular study, we had the data. So we wanted to investigate how well different labs, which are part of this core facilities association, how well they can find proteins which are in the sample, and how well they can distinguish which proteins change in abundance between conditions. And the focus was really more on the statistical aspects and not on the data collection aspects. And so for this, we needed two things. So one was where every participant of the study would receive the same data, so that the difference in results would really be due to data analysis as opposed to data collection. And we also needed to have a framework where we know the truth, right? Because if somebody finds one answer and somebody else finds another answer, we need to know, well, what's the right answer, right? And so what we did for that was we created a controlled mixture. And by the way, statisticians really like controlled mixtures because it really does have a sense of the ground truth. So in this particular case, it was a tryptic digest of yeast. So this is a complex background. And this background, we had these proteins spiked in different concentrations. So in mixture one, we had these concentrations of the proteins spiked in the sample. In the next mixture, we had some other concentrations, and so on. And so now what we can see is that the background proteins would be in constant concentration throughout. And in the spiked proteins between, for example, mixture 1 and 2, there will be some change, right? Between 1 and 3, there will be some change, and so on. And just as an aside, this part of the spiked mixture is actually called a Latin square design. So this is a design where each protein is present in the sample in a different and unique concentration, and then in each mixture it is also present in different concentrations. So this has a nice additional benefit that at least for these proteins the total amount of protein is constant. And then they added also additional proteins here. So this was the design. So we had each of the 4 mixtures, and they were acquired in replicates. So a little bit more details on the experimental procedures. So there was tryptic digest. So 3 technical replicates per mixture. The order was randomized. It was run on thermal instrument in data-dependent acquisition, right? So this was label-free, DDA. There was some technical parameters here which are all documented. And so then what we also did was we made available raw data to the participants, but we also did some partial processing. So for example, one step was where we just identified MS-MS spectra which correspond to MS1 spectra. So we did database search, used some ways to control false discovery rate and identification. So we found 5,700 proteins in these mixtures. So 48% of proteins had very few peptide spectrum matches, 1 or 2, and 29% of proteins were quite abundant, so they had 15 or more PSM. So this was one step which could be done, and then the next step was we also, in addition to identification, we also provided MS1 quantification with Skyline. And so essentially, now we have a situation where we have controlled mixtures with known proteins that do not change in abundance, known proteins that change in abundance. We have replicates, and the participants could start either from raw data, or from identifications, or from quantifications. What we didn't tell them is which protein was actually differentially abundant. So we essentially created some fake yeast-looking protein names for those, and we said, okay, now you need to take this data, start from your favorite point, and tell us which proteins are differentially abundant between each pair of those 4 mixtures, and which proteins are not differentially abundant.
Can we ask something now?
Sure. Oh, almost definitely. Please do.
The Q value and the but you've done no filtering of the data.
Well,
You do every year, right?
so yeah, there was some filtering. I don't have all the details here, so let me maybe give you the upshot. So the study and the data set is actually publicly available, and like all the like little detailed steps of data processing and so on is documented in this manuscript. So essentially, we did our best here, right? And in fact, we had people who were involved in the development of MSGF Plus, who, you know, we had people from Skyline and so on. So we did our very best as a group trying to find identifications, and that's what's available to the participants.
What is the difference in Q values RGB4 and the
Say this again?
Just want to know the difference.
Between the Q value and the false discovery rate. So false discovery rate was controlled at some level, which I don't have noted here. That's the problem, right? But it was definitely controlled for false discovery rate. However, this was not prescribed. So what the point was that we start from the data as it comes, right, or different intermediate steps, and then the participants could use whatever they wanted to tell us essentially yes or no. Each protein changes in abundance or not, and also report things such as fold changes, right? And then maybe if they have any statistical measures of uncertainty, provide those as well. So we did not prescribe any kind of particular metric for that because we wanted to see what people would come up with, right? And so yeah, that was the idea. So just very briefly, we had all kinds of people coming from different continents, had different job descriptions, coming from different facilities. So but many were actually, so you see that the majority came from the core facilities and doing some type of research. The majority had more than 10 years of experience in the field, but some had less than 1 year experience. Many were bioinformaticians, but others had some other type of roles. Many Europeans, but for some reason. So here we go. So now this is the interesting part, right? So everybody submitted their results to us, and it was de-identified, so we didn't know who were the participants. So we just had these identifiers of the participants, and they also had to fill, so first of all, they had to tell us for each protein that they identified yes or no, differentially abundant or not. And then they also had to fill a very long questionnaire saying we did this step, and then this step, and this step, and so on. So this slide summarizes the results of this study. So here the x-axis are the participants, and the y-axis is the number of proteins reported by the study. This group used intensity-based quantification, so MS1-based quantification. This group used spectral counting, so we didn't impose any constraints. This was some kind of hybrid. These guys didn't say anything. Okay. Now, the color is the type of input data that they used. So, for example, the brown here, this was the MS1 quantification that we provided to them, right? So they all have the same number of proteins because that's what they gave to them. But the various flavors of yellow colors, these are participants who started from raw data, and they used their own internal workflows. And you see already that there's a great variability, even in the number of proteins identified, reported by the studies. Well, these misunderstood the instructions. They only reported differentially abundant proteins. That's part of the process. So you see already that even by the number, here the study reported more than 6,000 proteins from this mixture. So even by the number already, if we look at the identifications across the studies, there's quite a bit of difference in terms of what is reported. Okay, so, but then we're interested in actual changes, right? So now this is the slide where, again, the x-axis is the study. And these are intensity-based quantifications, these are spectral counting, and these are the different methods here. The bars below the horizontal line are the true positives. So the participants didn't know what was true, but we knew, right? So for each protein, they could tell us yes or no, changing or not changing. So 4 choose 2 is 36 possible changes. And so many studies got almost close to the maximum, right? The bars above the horizontal line, these are the false positives. So these are the background proteins in yeast which were claimed by the participants as changing. And so you see that there is a great diversity in terms of how well different groups performed. And so here you see the triangles, they went above the limit of the y-axis. So this group found 2,000 differentially abundant proteins in the spike in mixture. And again, you see that the colors kind of mix together, so it doesn't matter if they started with raw data or with peak intensities. Some of them did well regardless of the starting point, and some of them did not so well regardless of the starting point. And you could do well or not well with intensity, or with spectral counts, or with hybrid methods, and so on. And remember, these are the same files, right? So there's no difference in data collection. The only difference in this performance is in data analysis, right? Okay. And so then we can look at other things as well. So we know what are the background proteins. And the background proteins are not supposed to be changing. So we also looked at the estimates of fold changes reported by the participants on absolute value, right? And so here again are the studies, and they're ordered in the same way as the studies on this slide. So these are the studies ordered the same way. And the box plots, they are the estimates of log fold changes across all the proteins that each participant identified. And so what you see is that there is quite a bit of variability in fold changes among the background proteins. However, these participants were not misled by some of the large fold changes, and they managed to distinguish noise from the systematic variation. But these participants were clearly misled by some of the changes in abundance, and they thought that they were systematic. And again, same data files, right? And so this tells us the importance of doing very careful work with data analysis, because it propagates itself all the way to essentially our biology, right, to the biological conclusions that we make. And this holds across the board with spectral counting, right, and with other methods. So, well, as I mentioned earlier, the participants also had to fill in a questionnaire with describing what type of things they did. It turned out to be very difficult for us to parse this questionnaire, because on one hand we didn't want to be prescriptive. We didn't say, okay, tell us what your p-value is, because it would imply that they have to report a p-value, right? So we didn't want to force it. Maybe they used some Bayesian method with some posterior probabilities, you know, or something. So the questionnaire was fairly flexible, and so we tried to group different things kind of by type of steps, right? So for example, the first column here is the input, right? So the raw peaks, raw data or peak intensities and so on. The second column is the methods they used for identification. So there are options like Skyline, MaxQuant, Progenesis, and some others. The green column is what they used for quantification, so feature intensity, spectral counts. The next column is how they summarize this, for example, at the peptide level, at the protein level, and so on. The pink column here is the statistical software, so I don't know, R, Excel, or Perseus, for example, with MaxQuant, and so on. And then the blue column was the linear, like the statistical analysis, like t-test, ANOVA, or something else. And the purple column is what they did for multiple testing. Did they use, you know, specific methods or not? So I really don't expect you to read any of that. My point is that there are many colors on this figure, right? Meaning that there are many, many ways that these people went about the same problem, starting from the same data point. We tried to do things like clustering, right, or something like that. Nothing came up. So the only real kind of thing that we could distinguish between them is according to the positive predictive value. So positive predictive value is the percentage of true changes among the reported changes. And so you see that this group did what we think is fairly well. So it's 70% of changes are correct. Here, the group between 20 and 70% of reported changes is correct, and this is a really bad, less than 20% of reported changes are correct. So that's really the situation. And so when we did all of that, of course, well, you know what happens next, right? So people will come and say, okay, so did MaxQuant do better than Skyline, right? Or did my algorithm do better than somebody else's algorithm, right? So of course, people wanted to look into that and kind of make some conclusions along these lines, so we did look into that. And so here's some examples. So these are the participants which all used MaxQuant and Perseus. And so you see that some did very well, some did less well. So this was the group which used Skyline and some linear modeling in R. Some did well, some didn't do very well. So the same thing kind of holds when comparing peak intensity versus spectral count. So these participants used similar workflows, but peak intensity and spectral counts found that both did well. This one workflow does much better than another. So even comparing this type of things can be very, very different. So the only thing which really came across very consistently in this is the following. So what I do here is that this column in the heat map is the identifier of the lab. In fact, the labs could have more than one submission. They were welcome to try many different things and submit that. And so what I do here, I circle the submissions from the same lab. And so if you see that, just with one exception, the circles never cross this line. So what this means is that the labs who know what they're doing, they can use multiple tools and they will do fine. And the labs that need training, they will use multiple tools and they will not do very well. So this was really the main conclusion for us, that this is really not as much about the tools as about the full workflow, which in this particular case, really data analysis, but as a statistician, I would like to argue that it's more than data analysis, it's also experimental design and your approach to data collection and then data analysis.
Do you have it on years of experience? Where does that break down?
It's actually a good question. I didn't look into that. Maybe there is, but it's also hard to know because sometimes these are team submissions, right? So it's hard to know exactly what is that. So we really look just at the consistency of their results, right? And again, years obviously help, right? But sometimes you also need to make sure that you can be a young person doing things right? So. And this is why we're here, right? So this was really a big motivation for us, that it's not just about the tools. It's not enough to learn OpenMS. It's not enough to learn Skyline. There is more that goes into that to do reproducible research. And that's really a big motivation for that. So just a few more things. So in terms of the type of things that we will do, so not necessarily this week, but starting next week for sure. So we will be looking at things such as how to actually distinguish systematic changes between conditions from random artifacts, and we can generate plots like that, where the x-axis is the fold change on the log scale, the y-axis is some type of statistical significance. So in this case, high values on the y-axis mean statistical significance, low values mean no statistical significance. So this is minus log p-value. So essentially, the horizontal cutoff is statistical significance. We can also use vertical cutoffs and fold change as a practical significance, so we will do things like that. And what also happened, actually, from this study is that once, it took a while to do all of that, right, to have the submissions and then analyze and so on. And since then, the tools evolved. And in particular, we processed, we did MS1 quantification with Skyline. So a year later, Skyline evolved, right? So we found new ways to do things, and so we reprocessed the same data once again with Skyline. So what we found is that, so here we have the true log fold changes, and the y-axis is the reported log fold changes. So on one hand, we can compare things such as spectral counts versus peak intensities. On the other hand, this was the previous year, and this is the year after changes, and we can see how the kind of consistency of quantification is improved with the correct data processing. So my point is that even though for this 2.5 days we will focus more kind of on statistics, the data that goes into statistics definitely matters, right? And so that was the focus of the beginning of the week. And then the same thing, so this was for the spiked proteins, right? These are the true changes versus log fold changes, and these are for the log fold changes among the background proteins. So spectral counts are very nice because they have a signal compression, but this was, so the median of log fold changes with old processing was 0.1. It went down with the new processing, so clearly as the tools improve, that of course helps. And what we will do at the end of the next week, just for those of you who stay with us, is actually looking at the influence of different decisions that we make as part of working with Skyline or OpenMS or any other tool, and how it propagates itself to the statistical conclusions. So here is some examples from the IPRG study. So here we only looked at proteins which have at least 2 peptides, and the data for each peptide will include the sum of 3 isotopic peaks. And so we also looked at different Q-value cutoffs, and we look at the positive predictive value among those. And then what happens if we only work with monoisotopic peak and not 3 isotopic peaks, how it changes the positive predictive value. Obviously, it's worse, right? And then what happens if we include single hit proteins, and we also, in this case, some 3 isotopic peaks. Again, we can look at the positive predictive value. So essentially, these decisions, we can watch them being propagated to the statistical conclusion. So this is the plan for the next week. And as I mentioned, so the data are available, and we will actually work with this data as part of the hands-on exercise. This was published last year, I think. So that is the motivation. That is the motivation for that. Any comments on that so far? Okay. So now I misplaced the slide, so I'll just go all the way back to kind of now we will go into the statistical methods, right? So since we're here, I hope we can all know that it's important, right? But let's kind of review why statistical methods are actually so important for our field. And the fact is that the data we're working with have a lot of variation and uncertainty, which are unavoidable. And there are many sources of variation. So there is technical variation in the whole process, such as where the samples come from, how they're handled, how they're stored, how they're processed, for example, in terms of enrichment or digestion and things like that. The instrumental variation, variation in retention time, variations in ion suppression, and so on. So this all introduces variability in the reported intensities. Signal processing, as you have seen just now, right? So depending on the tools that were used and the decisions that these tools make, they introduce also differences between the reported values. However, all of that can be solvable, at least in part, if we are very careful in our protocols and all of that, right? But this is not the whole story. And the reason why it is not the whole story is because the main objective of everything we do is that we study living systems. We study living organisms which have a substantial amount of biological variation in the abundance of proteins or peptides that we're interested in. So from one person to another, we have natural variation of protein abundance. Within the same person, from one day to another, we have natural variation of protein abundance. And so even if our protocols are optimized to perfection and we have no kind of room for variability, it will never happen, but even if it could happen, we still have to deal with the biological variation. We have to have a way to distinguish natural biological variation from the systematic change which is induced by disease or treatment or stress or whatever we are studying. And therefore, we need to have tools which will say, okay, how much natural variation is there? How much variation we see between different conditions? And is this variation larger than what can be expected by random chance? And so this is where statistics comes in place. And you can think of statistics as one definition as a body of methods for making wise decisions in the face of uncertainty. So this is one way to put it. And so the way statisticians contribute to this is really in 3 ways. And I hope that we will really kind of get to see all of these aspects in these 2 weeks. So the first is experimental design, and this is the focus of this hour, is that we need to design the experiments which really help us answer the question which was answered, as opposed to some other question. And at the same time, also help us answer a question in an effective way. So, somebody used to tell me, I don't like to work with statisticians because they ask for replicates, which is true. We'll have to ask for replicates, but at the same time, we can design effective experiments which will maximize the information content in the data given the same sample size. And this is very important. And so the second is data analysis, right? So now that we have the data, how we distinguish random variation from the systematic changes. And the third thing is statistical software. And it is only useful when the methods which are implemented in the software are sound and when the methods are used correctly. But at the same time, being able to automate things is extremely important, not only because it takes time to work with the data in Excel. The main reason is that we can repeat and reproduce what we try to accomplish. And this is really important. And so if everything is automated and programmable, then we can actually go back to the previous analysis. Like I have a friend who says, my worst collaborator is myself 3 months ago, doesn't answer emails, so you don't remember what you did. So if you have a documented script that does everything, you can reproduce your work much easier. So that's the idea. Okay. So now let's kind of see what exactly it means for mass spectrometry and proteomics. So a lot of the statistical practices like that, right? So we say, okay, we'll do that, and then, oh, let's use this method, and then let's use this method, and so on. And so really our goal is kind of untangle that and say, okay, let's see if we can have a predefined protocol which will allow us to go straight to the answers which are as reproducible and as meaningful as possible. So what does it mean for us? So from the statistician's perspective, there are really a lot of things that have to happen both before and after acquiring the data. So you may have seen, like in some talks, experimentalists, they would say, we have this really important sample processing step and really important data acquisition step, and then they have a little box, and then we have data analysis, right? So in my case, it's the opposite, right? I have all the statistical steps, and I have all the statistical steps, and a very little box in the middle data analysis. So not to minimize the importance, right? But that's how statisticians think. So a lot needs to happen before we collect the data. And so what I would like to do is to really focus on the experimental design in the beginning, and then also once we kind of work through the data, at the end of this week we'll come back to the experimental design, given what we learned in terms of the methods, how we can improve the experimental design. So this would be our goal. Okay, so the very first step of experimental design is actually not designing the experiment itself, but the very first step is to clarify your question. And maybe it's not your case, but it's definitely my case, where I have collaborators who come to me, and if you just try to kind of dig a little bit, you realize that they don't have a very clear question in mind. So they may say something like, I have these really valuable samples, and I just want to run them and see what happens. And I can tell you up front, nothing will happen. So it's really important to have a very specific question, and this question on one hand is a scientific question, right? So it's the science. So what exactly we're trying to learn from this type of samples, right, or from this type of problem. But the second thing is, how can we translate the scientific question into a statistical question? And this is super important, because if we translate this in a statistical question correctly, we can then best match the question to the statistical methods. And this is unfortunately very common, that people use methods which are not appropriate for the question. And so before we even go any further and we design the experiment, so I really want to make sure that we are clear about how different scientific goals can be matched to different statistical goals. Okay, so we can think of this broadly in terms of 3 types of goals of statistical analysis. So the first statistical goal is class discovery, where essentially we have data and we're looking for patterns. And you all have seen things like that, where there would be a heat map, where the row is, let's say, a protein, and the column is a biological sample, and the color is the abundance of the protein in this sample. And so we would be interested in in proteins with similar abundances across samples, because then maybe these proteins are somehow involved in the same biological process. Or maybe we would be interested in the samples which have similar profiles across proteins. Maybe this is some study of disease, and maybe these samples have the same disease subtype or something like that. So this is also quite useful. And the methods people would use for this will be various types of clustering. For example, here, the x-axis will be the sample, the y-axis is the abundance of the protein, and so we can see how we have similar profiles. Or we have methods such as principal component analysis, where each axis is some linear combination of all the proteins for a sample. Each dot is a sample, and if they cluster together, maybe they kind of are similar in this highly multivariate space. So this is good, and this is really good for things such as visualizing the data. This is good for things such as quality control. For example, if one sample was clearly compromised, it would probably be standing out very differently from the rest of the sample. So this is good to know. the mistake that people make, sometimes they say, oh, look, I have a PCA, and I have my, let's say these colors are disease groups, therefore I found biomarkers of disease. This type of objective, finding patterns, has nothing in common with biomarkers of disease. And the reason for that is that this method has does not take as input any information of disease or any kind of class like treatment and no treatment, knockout, wild type, or anything like that. It looks for patterns. So you say, go and find me something that looks similar. It will go and find you something that looks similar. How useful it is to you, it's a different question. But it will be similar, right? And so if you look for similar things, you can find things like that, where the divorce rate in Maine correlates with per capita consumption of margarine, right? Because there's some underlying causes, right, which have nothing to do with our question, right? And so this can happen. And so it is very nice to look for patterns, but there's absolutely no guarantee that these will be patterns which are useful to some labels of the samples or labels of the analytes that you may be interested in. However, these are good for visualizing the data or for exploratory kind of study of patterns. Okay, so this is one statistical goal. Then there is the next statistical goal, which we will actually spend quite a bit of time on this week and, in fact, the next week as well. This is probably most common in many proteomic applications where we are interested in comparing groups. So in the previous case, it was an unsupervised method because we didn't give as input to the method any information about group, healthy or disease or anything like that. In this case, we'll actually say, okay, I have some healthy people and I have some diseased people. And by the way, I will speak about healthy and disease just because it's so easy, right? But you can kind of translate this to any additional context you are interested in. And so let's say we have some healthy people and we have log abundance of a protein. And by the way, we will talk about log abundance a lot, and I will clarify the reasons in a little bit. And so here is the abundance of the protein of the disease. And so this particular method will ask a question, is the average of protein abundance in a healthy group systematically different from the average protein abundance in the disease group? In other words, is the protein upregulated or downregulated on average among these subjects? And the methods you would use would be something like t-test or analysis of variance, or so you have certainly heard that. So this is useful, and this is a supervised method, and it would report metrics such as p-value or q-value or posterior probability or something like that. However, there are multiple problems with that as well. So for example, if our groups do not correspond to the actual populations we're interested in to study, we can kind of end up with, let's see if I have something, something like that, right? So when we have, okay, yesterday you had 0 husbands, today you have 1 husband. If you can go like this, very soon you will have a dozen of husbands. Better get a bulk rate on the cake. So if you try to study a subset of the population, but your hope is to extrapolate somewhere else, it would really not make any difference. So this is one problem. But another problem is that, let's say again, going back to the biomarkers of disease, a common mistake that I see is that people will have, let's say, a t-test between 2 groups, and they will have proteins with small p-values, and then I will see in the paper that we found biomarkers of disease, which are proteins with small p-values. And again, I would like to claim that proteins with small p-values have very little to do with biomarkers. And this is a very common mistake as well. And this has something to do with the fact that the statistical question doesn't match the clinical or the biological question. And here is the reason. When we ask the question statistically, we are interested in comparing the average protein abundance in a healthy group to the average protein abundance in a disease group. Biomarker, on the other hand, is supposed to be actionable for an individual person that is in front of the doctor. So the doctor doesn't see an average person, the doctor sees a particular person, right? And it's okay if on average this protein is changing. For this particular person, it may or may not be changing. And so here are some examples. So we can have, let's say, 2 groups the proteins are healthy versus disease, right? So let's say we have the means of the two groups, and the proteins is very tightly regulated in each group. So clearly, if you have a person who comes with this protein abundance here, it's a disease group. If the person comes with this disease protein abundance, it's a healthy group. So this will be a protein which will have a small p-value. It will be differentially abundant, and it may be predictive of the status of the disease for an individual person. But here's the situation when you have exactly the same difference between the means, but now the protein has a lot of variation in each group. And if you have a large enough sample size, you may be able to have a small p-value, but if a person comes with this value of protein abundance, it can be healthy, it can be disease. So a small p-value will not guarantee that this protein is actually predictive. So in this case, the statistical method which compared averages does not match the clinical question, which is looking for the prediction for an individual person. And so it's a mismatch, and it's not the right choice. And here, by the way, is the opposite, right? So if we have, let's say, two proteins, and the blue dots are healthy subjects and the red dots are the disease subjects. So here, the proteins, both proteins, are both differentially abundant and predictive, right? If you have low values, you're healthy. If you have high values, it's disease, right? But here's a situation where, let's say, the protein 1 spans the full range of abundances among both healthy and disease. If you try to compare averages of protein 1 in the healthy or in the disease, they will be very similar. Same thing for protein 2. But as a multivariate panel of two proteins, they're perfectly predictive. So this is a situation when we have a predictive panel, but the proteins do not have small p-values. So it's really different questions and different methods. And so we need to have different method which is, aligned with the goal of the experiment. And so the third statistical goal is that. So the goal is class prediction, and this is really the type of things that would be useful if you are interested in predicting the condition or the status of an individual biological sample. But this is an overkill if all you want to know is if your yeast samples have a protein upregulated or downregulated, right? So in this particular case, again, it's a supervised method because the method, if it tries to discriminate healthy from disease, you need to give it examples of healthy and examples of disease. So each sample is labeled. And so let's say, again, blue is healthy and red are disease subjects. So we have two proteins. Here will be a protein panel which essentially ignores protein 1, and it says large values of protein 2 means healthy, low values of protein 2 means disease. And so we just have a cutoff, and this is mainly kind of markers function like that. But you see that we have some misclassified points. So then we can say maybe we can do something better. Maybe we can have a bivariate or like panel of two proteins, and now we have a curve which uses both protein 1 and protein 2. And so now we see that we misclassify fewer red dots. Or maybe we can have some really, really complicated rule which uses both proteins and describes perfectly each dot in our data set. But this may be a problem because maybe this accommodates too well the random variation in our data, and maybe it overfits our data. So maybe this is complex, this is maybe too simple, this is maybe just enough, this may be too complex. And so the methods that you would use would be essentially looking for some middle ground between something too simple and something too complex. And this will be machine learning methods, which would include logistic regression, support vector machine, random forest, deep learning, so methods like that. However, these methods do not answer the question, what is upregulated or downregulated on average? And so, again, the statistical methods have to be adapted to the scientific question at hand. And so that's a really important point for us. Oh, and of course, you can kind of overdo it as well. So here's our unique justified feature, uses deep learning to find data that agrees with your point of view, right? So you can also kind of, if you overdo it, right? So if you kind of go with methods which are too complicated for your data, it can also go wrong. Okay, so this is really the background for that. Any comments or questions so far? Nothing. Nothing at all? Okay then, so in this case, let us talk about experimental design, because this is really kind of the beginning part of what needs to be done for the experiments that we have in mind. So again, let me talk about healthy and disease just because it's easy, right? But you can translate this to your own situation, right? To any type of treatment or any type of problem. So when a collaborator comes to me with a potential project, so what I'm trying to show here is what actually happens in my mind when I try to translate the life sciences problem or subject matter problem into a statistical problem. So let's say we have a study of a disease, and so I would kind of project a situation where we have two populations of subjects which are identical for all things which can affect protein abundances, such as age, gender, other diseases, lifestyle, and whatever you want, except that one population are healthy and the other population has the disease. And this would be an example of statistical goal number 2, where we compare the averages between the two populations. So my question would be, on average in the healthy population, is the protein different in abundance from the average in a disease population? And so it may be useful for basic science. It may also even be useful for clinical applications where we're just in the beginning of kind of trying to find what are possible candidate proteins to follow up. Now, these populations are, for all practical purposes, are infinite. So we cannot go and investigate every single person in this population. So what do? We need to include in our study a subset of subjects from these corresponding populations. Now, as we said earlier, there is natural variation of protein abundances from one person to another. And so therefore, this is the first layer of random variation that we have in our study. Because if we want to reproduce our study, we will go back and have some other individuals, and they will have some other protein abundances. So we will not have exactly the same thing, but hopefully it's qualitatively it will be the same thing. So the first layer of variation is actually biological variation, which comes from the fact that we select individuals in the study randomly. But this is not the whole story, because at least for the moment, our measurement tools themselves, they have some technical variation, right? It comes from data sample processing, data collection, data processing, and so on. And so even though we selected some subjects into the study, we still don't know what their protein abundances are. What we do see is peak intensities, which are noisy readouts of a subset of subjects from the population. So we have two layers of variation, right? And so one is the biological variation, and the other is the technological variation. And what we're actually trying to accomplish is to take what we see. So the only thing that we actually measure is this noisy measurements on selected individuals. And so we try to take this as the input, and from this make conclusions about what we actually want to know. These are the underlying populations. So this is a process which is called statistical inference. So take the data that we see, so take what we observe, and make conclusions about what we do not observe, but what we actually want to know. And because we will make conclusions about what we don't see, it can go wrong in two ways. And these are the two dangers of statistical inference. So the first is the bias. And the bias comes when essentially our conclusions are systematically wrong and tell us systematically different results from what is actually happening in the populations. And bias is the worst, because bias is reproducible. Because now if you do the same thing again, you'll have the same mistake, you will see the same result, and you will say, great, I reproduced my results. Well, guess what? You are reproducibly wrong. So one important issue of the experimental design is making sure that we eliminate bias. The second aspect of experimental design is inefficiency, where the data that we collect essentially have so much noise and variation that we cannot say anything useful about the underlying populations. And so the data there, maybe they're not systematically wrong, but they just do not contain enough signal to actually make reasonable conclusions about the underlying populations. And so this is where statistical experimental design really plays in, making sure that we avoid bias and we minimize inefficiencies. So I won't have many formulas, but a little bit, just to again help you see how statisticians think in this context. So usually, traditionally in statistics, we use Latin letters for what we observe and Greek letters for what we do not observe. So in this case, we use Greek letters to say that essentially out there in the population, there exists a quantity, which is the mean abundance of the protein in the entire population. We don't know what it is. We will never know, never ever, right? But it's out there and it exists. So this is the quantity of interest. And the same thing for the second population. And so our quantity of interest now is to understand the difference between mu1 and mu2. So this is essentially a number which exists as unknown that we want to study. Now, what we do, we select the subjects, right, as we just said. So now these subjects have their own protein abundance. And again, we don't know what it is, but it's again numbers which are out there, they exist. And now we have as many numbers as subjects, right, in the same group, as many numbers as subjects. But again, they're unknown. What we do see is peak intensities. And now we have even more numbers, because for the same protein we can have multiple peptides, right? We can have multiple transitions or multiple fragments, right? And so on. And so this is what we see, and it has two layers of variation, the biological variation and the technological variation. And the goal of statistical inference is to create some kind of summaries from what we observe, characterize their statistical properties, and essentially use the statistical properties to make conclusions about the difference between the two constants. And so in this case, bias means that our summaries are systematically different from the true differences, and the efficiency means that the variance of these quantities is too large. And so this is how statisticians will go about this. They will say, how can we design the experiment to make sure that these numbers are as close as possible to these numbers, and this variance is as small as possible. So this is really kind of the whole mindset for that. Okay, and so this can be overcome by the three fundamental principles of experimental design, which we will now talk about. And I did not invent them. They were invented way over a century ago, right? But they apply very well for everything that we need to do. So before I move on to that, let me just tell you briefly why we talk about differences and not about ratios. So you probably heard a lot in the previous 2.5 days about ratios of various things, right? And analytical chemists love ratios, right? So we can kind of see. And the reason for that is that oftentimes biological signals and even technological signals, they're multiplicative, right? So that things increase in folds, right? And this is a very common situation. Ratios have horrible statistical properties. So when I say we need to describe the properties here and make conclusions for that, ratios are really difficult to deal with. On the other hand, if we take the log of a ratio, it's a difference of the log intensities, right? And so on the log scale, the multiplicative signals become additive signals. And this becomes much easier to deal with in terms of all kinds of statistical modeling. And so, in fact, for the next probably a good, I would say almost until the end of the next week, we will primarily speak in terms of log fold changes, right? However, keep in mind that this is nothing else than log of the ratios between conditions. And I will come back to this again. So it is really not different from what you do elsewhere, but because statistical properties are much easier to deal with, that's what we will do. Okay, so we now want to talk about the three principles of experimental design. So the first principle of experimental design is replication, and I really hope that I do not need to convince anybody that we need to have replicates, right? At least some replicates. So why is it important? It actually has two uses. One is that allows us to actually carry out the inference and distinguish what's systematic and what's random noise. And the other is it allows us to minimize inefficiency. So let's just take a look at that. So let's say we have a very simple experiment. Let's say LCMS, label-free, 1 peak per protein. So we have healthy person gives us 1 peak, and a diseased person gives us 1 peak. So this is our data, right? This is healthy and disease. This is the value in the healthy person. This is the value in the diseased person. Is it systematic or is it random? Who knows, right? That's exactly the point, right? There is no way to know if this is a systematic change or is it an artifact of random variation. So to know if it's systematic or random, we need to know how much random variation was there. So here, for example, again, we have very tightly regulated proteins in the samples. So this change is likely to be systematic, right, and likely to be reproducible. Exactly the same difference, but now we have a lot of variability within each group. This is much more likely to be an artifact of random variation. So the consequence of that is, by the way, that fold change cutoffs make no sense whatsoever, right, for looking for differences between groups. So because exactly the same log-fold change here can give us a systematic signal, or it can give us a artifact. And I would probably think that many of the participants of this IPRG study that we talked about earlier, who really were misled by changes in variation in the background proteins, did something which some version of a fold change cutoff. So they really didn't do a very good job characterizing the extent of variability in each sample. So that is one thing, right? So we need replicates at least to understand how much variation we have in each condition. But the other thing is that, well, more replicates, it's true, allow us to be more confident in the mean. So if now instead of 4 replicates I would have 100, I would be really, really confident in the mean of the 100 values, right? And I would be really, really confident in the mean of the other 100 values. And maybe I can even claim that among 100 people, this small change is unlikely to be an artifact. So usually, larger sample size allows you to claim with larger confidence even small changes. So this is another thing to consider. So now, as we were saying, it allows us to characterize the variation. So we said that there are 2 sources of variation. There is a biological variation and there is a technical variation. And therefore, we can have 2 types of replicates, right? We can have biological replicates and we can have technical replicates. And how many and which we need, we'll have to come back to this at the very end of 2.5 days, because we actually need to know a lot about our whole workflow to answer this question, including how we're going to analyze the data. But let me give you an upshot right away. Biological replicates are a lot more important than technical replicates. So if you have 10 mass spectrometer runs possible, you will be much better off having 10 different people in your experiment than having one person analyzed really, really, really well, but it's still one person, right? And we will come back to this at the end of the 2.5 days. Does it make sense? Okay. All right, so this is for the replication. Now, the second thing is a little bit less obvious, although I hope that you know about that, but in practice, I see how often experimentalists forget about that, is the randomization. So the purpose of randomization is to prevent bias. And as I was saying, bias is the worst thing because, you know, if you have an inefficient experiment, you don't find anything, you say, oh, well, right? But if you have a bias and you have conclusions which are really in the wrong direction, you can actually spend a lot of money following up on conclusions which are really invalid. So it is really important to try to minimize biases as much as possible. So let's take a look at the example. So let's say we have, again, LCMS experiment, label-free, 1 feature per protein, right, per person. So let's say we have the experiment which has healthy subjects and the disease subjects. All the healthy subjects were acquired on day 1 and day 2 of the experiment. And the disease were acquired on day 3 and day 4 of the experiment. And so now we have this really large fold change, which is likely to be a systematic, like not a random artifact, because it's a really big difference here. However, this difference can be due to at least 2 things, right? So one is due to the disease, and the other due to the day of sample acquisition. And day of sample acquisition is really a proxy for many, many, many things, right? For many things which can happen to our experiment, including material change, weather change, operator change, you know, instrument problems, and whatever, you name it, right? And from experience, if I see this, it is much more likely that the change is due to the day than it is due to the disease. So in statistical language, it is called confounding, meaning that you cannot distinguish the impact of these 2 sources of variation. And in practical terms, it means that you wasted your money. And there is no amount of statistical sophistication which can help us disentangle these 2 types of things. So a better way to do that is to randomize completely our samples across all the days. And especially when the experiment has a large enough sample size, if we completely randomly select which sample is acquired on which day, on average, the days will average themselves out over the conditions. So what happens now is that healthy have roughly all the days present, disease have roughly all the days present, so now all the days are present, so there is no difference between the groups which is due to the day. The problem is that we now have more variability here, because now the variation within the group is due to two things. It's due to the disease, but it's also due to the day-to-day variation. So this would be an example of an experiment which is not very efficient, but at least it has no bias, and this is a really good start. And so again, because we have two types of randomness, we have biological variation and technological variation, we also will have two types of randomization, right? So we need to randomize the selection of subjects from the populations. And we also need to randomly allocate the samples at all steps of data acquisition, data processing, sample processing and data acquisition. Does it make sense? Right? So you may say, oh, wait, but I know which days, maybe I can do things, I can normalize and so on, right? And we will talk about this later. Randomization is really important, in particular, to protect ourselves against sources of bias which we don't know about. Because if we don't know about, there's really no way we can control for it. And there are really many ways where we can have artifacts in our data which come from sources that are unknown. And so randomization is a really key step. And I can tell you I had many cases where we had collaborators who forgot to randomize or didn't do it appropriately and so on. We had to redo the experiments. The results were really, really different. So we should not say, yeah, I know we should do it, but we couldn't because it's hard and so on, right? So it's really important. And by the way, in the hands-on parts, when we will be using R, one of the important things would be to see how we can do randomization in R. Because humans are actually really bad at randomizing. Actually, we have a few minutes. Can we make an experiment? I'm just curious, to see how good we can be at randomization. So let me write some numbers here. So let's say I have 10 numbers. So I have 0, 1, 2, 3, 4, 5, 6, 7, 8. Two numbers. So now, don't say anything, each of you. Think of a random number from this list. Okay? Right, let's have a show of hands. How many have 0? Okay. How many have 1? Okay. How many have 2? How many have 3? How many have 4? How many have 5? 4. How many have 6? 5, 6, 7, 8. How many have 7? How many have 8? Doesn't look like an equal distribution. So humans are terrible at randomizing. So when you say, I randomized my data by shuffling, the material, very bad idea. And you have so many examples. Like, for example, I read somewhere they were studying rats for some kind of behavioral treatment. And so they had two groups, so they had some rats with the treatment and the others were just controls. And so what they did, okay, let's just randomize. So they grabbed some rats out of the cage, applied the treatment, and then the others were the controls. Well, it turns out the rats they could grab, they were slower. And so that obviously affected the behavioral factor. So don't do it by hand. So it's really important to, and R, but it doesn't have to be R, Excel, anything. You can generate random numbers from pretty much any source. So this is quite important. Okay, so this is for randomization. Oh, and actually some examples as well. So this is something I really appreciate. it's fairly old, but I still think it's a nice example. This is a paper from statisticians who used to work at MD Anderson Cancer Research Center, and they are doing a lot of essentially statistical, service for clinical collaborators. And so it's a very old study, but still kind of the same thing holds. Their collaborators were studying 5 types of cancer. So at that time it was a CELDI experiment. So they had 5 cancer groups, and the statisticians, they received their samples de-identified, so they didn't know which sample came from which group. So they did all the right things, normalized, picked, and so on. And then they did clustering, looking for patterns, as we just discussed. And instead of 5 groups, they found 6 subgroups. So they were super excited, thinking, okay, we have a new subgroup of the disease. But then what they did next was they have this x-axis is the same order of samples as here, but then the y-axis is the day when the samples were collected. And you see that the clustering matches perfectly the day of data collection. Now it turns out that the first 2 groups of disease were acquired on these 2 days. So now there's no way in the world you can believe that this is a reasonable biological conclusion. And so this paper has actually a whole series of essentially failed studies, and I really appreciate them doing this, because not every day you see people publishing failed results. I think it's a very useful example. Even though these are old data, it is still quite valid. Here's another example, also for randomization, and I think it's more recent. So it's 2007, although not that recent. So this comes from a blog by Jeff Lick, who is a quite well-known statistician and advocate for reproducible research. So this is a paper which appeared in Nature Genetics in 10 years ago, which studied gene expression between Asians and Europeans. And so microarrays, right? So gene expression study. And this paper found that 78% of genes are differentially abundant. 78% of genes are differentially abundant between Asians and Europeans. Right? So that's a lot of genes, right? So, but they, of course, they had also colleagues who were interested in the results, wanted to repeat it, so they looked through the data analysis, didn't find anything wrong, so everything was done kind of as it should be. So luckily that study was done a long time ago with Affymetrix chips, and Affymetrix chips have a timestamp in the corner. So it took all the way to go in the very raw data and to go in the Affy chips to realize that there was a 1-year difference between what they profiled in Asians and Europeans. Well, no wonder that many things will be changing, right? But this has nothing to do with the biology, right, and nothing to do with the ethnicity. So these are likely batch effects. And again, this is an example of a study which makes no sense whatsoever. I don't know what happened to this paper. I haven't followed up with it. But so it went through peer review, right? So, okay. So this is another example, right? And so this is one of my favorite quotes from Ronald Fisher, which says, to call in the statistician after the experiment is done may be no more than asking him to perform a post-mortem examination. He may be able to say what the experiment died of. Well, if nothing else makes sense to you, right, what I will be talking about, or others, and so on, if there is one message I really, really want you to come away with, is talk to a statistician before collecting the data and before starting the experiment. It is really important, because otherwise, we can be in these situations and there's not much you can do. Okay, so this is for randomization. And so the last principle of experimental design is blocking. And so this is probably the most difficult one. And I would say replication and randomization, you can do it yourself. It's really not a big deal. Blocking already requires quite a bit of expertise and also quite a bit of understanding of the sources of variation in the experiment. So let's take a look at that. So again, let's imagine LCMS experiment with 1 peak per protein per subject. And so, as we just said earlier, if we have a day-to-day effect, and if we know it, we cannot control it, but at least we know it exists. So then our variation in each group has really 2 sources, right? The biological variation, which we cannot do anything about, and then the technological day-to-day variation, which is not desirable. So, but the important key is that we need to know that it exists, right? That it's an important source. So a complete randomization essentially inflates the variance to the point that this signal becomes very unreliable, and this is an example of an inefficient experiment. So alternatively, what we can do is to acknowledge that we have a day-to-day variation, and then we can impose a restriction on randomization. So blocking is really a restriction on randomization, where we say, instead of randomly selecting the order of collecting or acquiring the data, on day one, we will enforce this to be one healthy person and one diseased person. So there is still randomization in the sense that we randomly select a healthy person and we randomly select a diseased person. But we impose a constraint that should be one of each. Then the next day, again, one randomly selected healthy, one randomly selected disease. And so now, on each day, we have an example of a healthy and disease. So this accomplishes two things. One is that if you have a complete randomization, what we said earlier is that on average days, overall days will average out across groups. It is true for large-scale experiments. If you only have 4 replicates, even by random chance, you can still end up with, 3 healthy on day 1 or something, right? Some patterns which are not quite balanced. So imposing a constraint guarantees that you have exactly the same number of healthy and disease in each group. So this is one thing, especially for studies with low sample size. But then the second thing that we can do is to say, well, now we can look at the differences within each day. And so if you see in my cartoon here, even though we have a lot of variation within the day, between days, within the day, the difference between healthy and disease always points in the same direction. And so this points to the fact that in this particular example, in the disease, the protein is upregulated compared to the healthy. So instead of having 8 data points, we ended up with fewer data points. So there are only 4 differences, but these differences are a lot more systematic. So this allows us essentially to eliminate the day-to-day variation, even though it exists and we cannot control it. So it requires 2 things, right? Design the experiment in a way that actually produces this type of blocking structure, and then the second is analyze the data set accordingly, right, to account for this difference. So now, you probably, oh yes, please.
So we only do that if the order is the same. For instance, the middle graph there, bottom to top, we've got on the left column healthy, day 1, day 2, day 4, day 3, and then the right hand.
Here, yeah.
Left hand bottom is 1, 2, 4, 3, 1, 2, 3, 4. So after 3 is going down.
Yeah, so I think what you're asking, let's see if I can show you. The things essentially cross like that, right, for 2 days. And that's totally possible. So of course my cartoon is an idealized version, right, of what's here. So I think what you're asking for is if I have a picture, so this is healthy, this is disease, and then I have something like that, right? And then you would have, I don't know, this and then this, right? Something like that.
If most of them are going one direction, but only one was going the other direction.
Exactly. So essentially what you are doing now is you are summarizing all these differences, and you are saying, are these differences on average 0? Or is the average tend to be systematically above 0 or below 0? There is absolutely no requirement that they all have to be going in the same direction. The point is now that you have a different input data, right? So you summarize the differences as opposed to the mean of this group and the mean of this group. And these differences, because they're all within the same day, they eliminate the day-to-day variation. And in fact, it may be kind of, the picture kind of not usual, but especially for analytical chemists, I think that's really very familiar, because remember, the differences, they are log ratios. Right? So looking at ratios of analytes, right, which come from the same system is very familiar. Now the problem with the ratios is that you can only do them with 2 numbers. And blocking allows us to do this with a much bigger kind of experiments. For example, in this case, I kind of pretend that I can only do 2 samples a day. What if I can do 4 samples a day? What if I have 2 healthy and 2 disease within the same day? So then it's not very clear how to calculate the ratios. Turns out the statistical methods can really integrate all of this information in a much more seamless way if you take the log and work with them on the an additive scale. And we will talk about that later. So this is one of the reasons why we want to work on the log scale. But that really is kind of essentially something which is very familiar, I think, for those who work with instruments frequently. But going back to kind of the experimental design, remember we were saying we have 2 layers of variation biological variation and technical variation. Therefore, we can actually block on these types of variations. So this was an example of the day-to-day variation. So blocking on the technical variation, we can also block on the biological variation. So let's take a look. So here's an example of what happens kind of in a similar situation from the same paper. So this was an example with 2 types of cancer. And again, they analyzed the data in the right ways. It was also de-identified. So they did the clustering, found 2 groups, were very happy. Well, guess what? So they found that the difference between the clusters was the protocol change, right? Which is obvious, right? If we knew what it was. The problem was, again, that before the protocol, they had all the, like, one group, and then after the protocol change, they had another group. There's no way these things are comparable, right? So these differences are all artifacts. So protocol change may be unavoidable. For example, if you have larger scale experiments, you need to change some materials in the middle, right? The operator change or something like that. So this would be one example of blocking where you make sure that before the change of protocol, if you know this in advance, right? Before the change of the protocol, you have the same number of healthy and disease. After the change of protocol, you have the same number of healthy and disease. You look at the differences here, you look at the differences here, you put them together. So this is quite possible to do. This was not what happened in this study. But now in terms of blocking with respect to biological risk factors, so remember I was saying that one of the good things about blocking is that it enforces the balance, right? That accidentally we would not end up with imbalanced selected subjects. So here's an example, right? So let's say we have health and disease populations, and they're quite diverse in terms of age, gender, and other things. So if we just randomly select subjects in the study, if we have a large sample size, they will all average out. If we have a small sample size, just by accident, we can still end up with having like all older males in the healthy group and younger females in the disease group or something like that. So blocking would allow us to say, okay, I want to make sure that with respect to factors which are likely to affect protein abundance, I want to make sure that they become equal across 2 conditions. So for example, it would mean I want to have one younger female from healthy and disease group. I want to have one older female in health and disease group, one younger male in health and disease, and so on. So now it will enforce the fact that the age and gender distribution is comparable between the 2 groups, so we cannot confuse change due to the disease to change due to age or gender. But we can also analyze essentially each subgroup separately and combine the results. And this would eliminate the extra variation which is induced by difference in age or differences in gender. And so, by the way, when I say analyze it separately and put together, it's actually not what happens. We have a model which represents all of the sources of variation. We don't actually do it in different steps. But that's really the insight behind that. Here's an example of what happened in one of the studies that we had a long time ago with our collaborators, just kind of an illustration of that. So this was a collaboration with the Munich Heart Center in Germany, and this is a very big center for cardiovascular disease, and they just had the routine protocol to collect plasma samples from every patient who came in with a problem. And so they essentially had a really big repository of plasma samples, and our collaborators, they felt that age and gender usually affect everything, so they wanted to make sure that we stratify by age and gender, so they came up with these 4 groups of age and gender, and so these were the number of samples they had for each combination of disease and confounding factors in the repository. And so if now they want to include a small subset of the samples for their investigation, the blocking would make sure that you have the same number of samples in each row. Now, it doesn't require that you have the same number of samples across rows, but within the row, you want to make sure that each disease group has the same number of older females, older males, and so on. So this is for the block randomization on that. And I would say that the very well-known and very familiar to you type of blocking is actually labeling. Right? And again, it's just different choice of vocabulary that statisticians are using, but it's actually something which is very familiar. So mass spectrometry runs clearly have variation in them, right? So from one run to another, we have a lot of variability. So one way to minimize this variation is to profile samples from multiple sources simultaneously. Then whatever ion suppression or whatever kind of retention time issues occur, they affect both healthy and disease in the same way. And so labeling can be used in many ways, of course, right? So what you have heard in the Skyline part was using labeled reference peptides, right? So I just kind of talk about that. So what this means is that you have, as before, one peptide per protein per person, but that now we also use heavy labeled peptides with the same peptide sequence but a mass shift, which has spiked in each sample in a constant concentration. So now this is important because the red and blue peaks, they have biological variation in them. But all the variation in those gray peaks would be only technology. And so this allows us to see how much technological variation is there, right? And so now this would be the representation of that. So this is, let's say, the healthy samples. These are the disease samples. And the blue dots, these are the peaks from the biological replicates. And the black dots, these are the peaks from the labeled reference peptides, right? And so if we didn't have labeled reference peptides, we would be comparing the mean of the blue dots to the mean of the red dots. And if there is enough variation, there is not much difference between the means, and it's really hard to see a signal. So this is, again, is an inefficient experiment. However, if we have a labeled reference peptide corresponding to each endogenous peptide, and it has variation, so we know now that this variation is due to the technology. And what we can do, we can consider the deviations of the endogenous peptides from the corresponding standard. And again, I'm talking about differences on the log scale, but these are essentially log ratios, right, of the endogenous peaks to the internal reference standard peptides. And so we can see that if here the deviations from the standards are systematically larger than in the healthy group, then it points to upregulation in the disease group, even though there's a lot of variation. So this also allows us to gain efficiency. Of course, there's a price, right, because the sample complexity increases and it's expensive and so on, but clearly this gains efficiency in the experiments. Or you can do other things such as SILAC, for example, or TMT, right, when you mix samples from different labels. Okay, so just kind of putting this together, so this is where we are, right? So everything we've done so far is just this, right? We talked about the experimental design, how we frame the question in a way that we can match the question, the statistical question, to the scientific question. We talked about how to randomize, block, and select replicates. We didn't talk yet about how many replicates we need, and this is because we actually need to do all of that before we can get back to the experimental design and talk about that. So this will be the topic for the last section. So a lot of the things that I will be talking about tomorrow is actually described in this really nice collection of very short vignettes in Nature Methods. So it's a series called Points of Significance, where they have vignettes dedicated to really very precise topics in statistics, and I will be using occasionally figures from these vignettes. So there's a lot more than what I'm showing here now, so make sure you can have access to that. Okay, so this is all I had for today. We have a couple minutes left if you have questions. Yes, please.
Think about the difficulty of getting all these things to work together. You know, like you analytical chemists, but you have biologists who are running biology experiments with mice. So then you have no control over, you can only influence, maybe not even influence the design of that experiment, and when they're going to take them down, and who takes them down, and all of these things before you ever touch the sample.
Right, and then statisticians come later and then they complain, right? So I think that the solution to that is to educate your collaborators, right? And build teams and build alliances where you can become on the same wavelength, at least in terms of what's important, right? And maybe it will not be perfect, but at least it will not be systematically wrong, right? So you may, not be able to block, but in terms of at least finding replicates and randomizing appropriately, it is really key because, one thing you can do you can argue in terms of dollar amounts, right? So how much money you will waste following up on leads which turned out to be wrong because the experiment was not randomized, right? And that helps usually.
I was looking about how mocking is not just at the sampling, it's also at the analytical part. If you're not all running all your healthy first and all your
Most definitely, most definitely.
everywhere, it's the same concept that applies.
Most definitely, and you know, sample processing like digestion, you know, and enrichment, or I don't know, fractionation and all of that, it has to be, absolutely, it is actually really important. And I have seen many examples where we had to redo the experiment and the answer had nothing to do with the initial version, which was not randomized. So it's really, really key. So this was kind of the statistical methods part. So what will happen next we'll have a break, and then we'll have a hands-on session, so where it will be also an intro, but a completely different thing. So doing hands-on R, because R is a really useful combination with statistical methods. On one hand, because you can get quite far with very little coding, and on the other hand, because you can create essentially a record of the analysis which can be documented and reproducible, but also integrated with other larger projects in the lab. And so what we will try to do is, today, it will be just intro to R for very beginning, but then little by little we will merge some of the things that I will be talking about, and we'll see how to do them in R, such as randomization, right, or some other types of things. And then on Monday, we will talk about more advanced methods, which are specifically, kind of more specifically adapted to mass spectrometry and proteomics in R. And so I hope that this will kind of set us up well for the next week. Yes, please.
So, like the number of, in the clinical projects, how many replicates we should have, additional replicates? That's always the challenge, right? So you do some power calculation, etc., to come with that number. Is there some recommendation you have how to deal with that thing?
We will talk about this next week if you're around. Well, the short answer is maybe. Right? But in practice, well, it depends on several things, right? It depends on what you want to do. So, for example, if your goal is biomarker discovery and prediction, you would need a different sample size than if you just looked at comparisons on average, right? Second thing is, it depends on how much biological and technological variation you have, right? So, for example, if your scope of the experiment is very narrow in terms of target population, it may be more homogeneous, right? On the other hand, if you expand this to very broad population, let's say many ethnicities, many age groups, and so on, you will, of course, need a larger sample size, right?
Does that also depend on the type of technology being
Very much
used,
so.
For example, TMT labeling or LFQ?
And it also depends on who is running the experiment, because the same workflow in different hands can give very different variation. So what it really means in practice is that you need to have a really good sense of how you're going to analyze your data, but also have a pilot study in your lab with your particular workflow, which would allow you to quantify the sources of variation in your hands. So it's actually not very helpful to take some data from the web, from collaborators, because it may be very different for you, for various reasons. And so usually what we advocate is actually an iterative approach, where you start small with pilot experiments, you learn the components of variation, and then you go from there, kind of plan larger scale studies. And by the way, things such as system suitability and, assay development is a very important part of that as well, right? So depending on how that is set up also affects your sample size. Yeah.
So on 4 samples, if you took one of the samples and said it's one of the replicates, and said each one had an outlier, how would you, is it fair to say that there's an outlier, or how do you do that?
It is difficult. So let's say 2 things. So ideally, you would not remove outliers unless this is for a very good reason, right? So you have to be really confident that essentially an outlier means a mistake, right? It shouldn't belong there, right? So either there is a measurement error, right, or a sample processing error or something, right? So you really need to have a good reason to believe that this is a mistake. And usually you do that by defining a protocol before collecting the data, by saying, I expect my retention times to be within this window. I expect to be, my metrics like this, right? And so you will use this particular metrics to claim that there's an outlier, right? So otherwise, especially if you have a small sample size, outlier may actually be trying to tell you something. So what this may mean is that you have, in fact, a much larger variability in your biological process than your data indicate. And this can be due, for example, because of bias in data collection. So my favorite example in terms of outliers is, imagine that you have extraterrestrial who tries to study height of humans, right? Doesn't know anything about humans, just tries to study humans, right? And so they arrive on Earth and they land in elementary school. And they say, yeah, well, most people are that high, that tall, right? And they have a couple of outliers, but they're outliers, right? So essentially what happens is that you had a very biased data collection, right? And the outliers actually try to indicate that you didn't sample your population fully, right? And by eliminating them, you may actually get a very wrong idea of what's going on. So unless you have good reasons, based on your protocol, based on your experience with the data collection, so unless you have good reasons to believe that this data are really a mistake, what it may mean likely that your sample size is not large enough to cover the full extent of variability. And so eliminating this based on some kind of ad hoc criteria saying, oh, my 3 points are together, point number 4 is outside, I'll remove it. It's extremely dangerous because you can essentially eliminate enough to show whatever you want, right? So you can always eliminate things you don't like, and then the data will behave like you would like them to do, but it's not necessarily reproducible.
In one case, the disease has more variability.
Well, but these are not outliers, right? It's just variation, and that you can model, that you can work with, right? But outliers mean individual points which stand outside of the bulk, right? And so either these are mistakes, and if you have predefined criteria for defining mistakes, go with that, or it's possible that they're actually trying to send you a message. Yeah. Anything else? Okay, well, I'm around if you have questions after that. Otherwise, let's take a break, and 3:30 Laurent will come in and we will get started with R and learn all about that.